Hi everyone. In this video, I'd like to go over the DNA extraction process. So DNA extraction is the first step of the DNA barcoding process. Before we get to the DNA barcode, we need to make sure we have a good amount of genomic DNA in the sample we are working with. Here you can see all the supplies we need to conduct DNA extraction, and you can find most of them at the DNA extraction station in the lab. For DNA extraction, we use QIAGEN D-NISI blood and tissue kit, and we follow the manufacturer's protocol. This DNA extraction protocol has two parts, which are typically done over the course of 2 days. At the end of the first part, we place the samples in the incubator overnight, and then on the next day, we take them out and we proceed to the second part. However, you can do the whole thing in 1 day. In this case, you need to make sure the samples stay in the incubator for at least 2-3 hours. That should be enough to complete lysis. Here you can see the supplies you need for day 1. For DNA extraction in general, you need 2 pipettes, P1000 and P200, and corresponding pipette tips. For day 1, however, you need only P200. So the goal for day 1 is to break down the cells and digest proteins for the maximum release of nucleic acids. If you have just one sample, you need only one microcentrifuge tube. In this tube, you combine 180 microliters of buffer ATL, a piece of tissue you work with, it can be a whole insect individual, and last you add 20 microliters of proteinase K. You need to vortex the microcentrifuge tube very well and place them in the incubator at 56 degrees overnight or for 2-3 hours. It might be a good idea to vortex the samples a few times during incubation. Here you can see the supplies you need for day 2. For day 2, you need 2 pipettes, P1000 and P200. I typically start with taking out all the buffers from QIAGEN box, and I put them in the order I need them to use. The only thing which doesn't come with the kit is 100% ethanol, which we have in the lab. First, we need to take the samples out from the incubator, and here I have 2 samples, and we need to vortex them very well. Next, I usually set the pipettes for 500 and 200 microliters respectively, since these are the only volumes we are using at this step. Following the protocol, we need to add 200 microliters of buffer AL to the sample mixtures, vortex the tubes, add 200 microliters of ethanol, and vortex again. Next, we need to pipette approximately 500 microliters of the sample mixture into a D-NISI mini-spin column placed in the collection tube. Both the columns and the collection tubes come with the extraction kit. We centrifuge the samples using a microcentrifuge, which is on the bench near the gel station. Make sure you have an even number of the samples and you distribute them evenly in the centrifuge. You can use an empty tube to balance the centrifuge if needed. We centrifuge the samples at 8,000 for 1 minute. The purpose of centrifuging is to get a concentrated DNA. After centrifuging is done, we need to discard the old collection tube and place the column in a new collection tube. During the whole process of DNA extraction, it's important to understand where the DNA is in your tube. For example, here, after centrifuging, the concentrated DNA will be located at the column membrane, so we must not discard the column. Next few steps are the washing steps. We use two buffers, AW1 and AW2. Just follow the protocols. These are the same centrifuging steps. Just make sure at step 19 to change the centrifuge setting to 14 thousandths and centrifuge the samples for 3 minutes. After the last centrifuging step, we need to place the columns not in a collection tube, but in a new 1.5 microliters microcentrifuge tube. In this tube, we will store the DNA at the end. So we need to label this tube with all the information we want to have for the sample. You might also remember that the DNA is still at the column membrane. So next, we need to add the elution buffer, buffer AE, which helps elute the DNA through the column membrane into the microcentrifuge tube. Again, follow the instructions and incubate the tube for 1 minute just on the table before you place the samples in the centrifuge. The purpose of this 1 minute incubation is to give the elution buffer some time to penetrate the column membrane so that the DNA is ready to elute during the centrifuging step. After the first centrifuging step, it's important to remember that the DNA is now in the microcentrifuge tube. This is why we do not discard this tube, unlike the collection tubes before. At step 27, it's recommended to repeat the centrifuging step with another 200 microliters of buffer AE to increase the yield of DNA. Once we are done, we can finally discard the column and close the microcentrifuge tube, which is now ready to be stored in the fridge. That's it. You can keep the samples in the fridge until you need them for PCR, or if you like, you can proceed immediately to PCR.